Hello, everyone. My name is Katalin Patanai, and today I will be talking about combining field and laboratory methods for the assessment of the trophic ecology of 4 co-occurring invasive mysid species. The study was done in Hungary, where these 4 species co-occur in the same location. As we all know, aquatic ecosystems experience multiple threats. These can be threats from invasive species or pollution and many other things. And freshwater ecosystems are particularly exposed to these threats because they're close to cities, migration route on rivers, etc. So overall, the community, for example, the trophic impact of invasive species is often unknown. So there are these mysid shrimp, as we call them. Here's an example of Lemna mysis benedeni, and they're one of the most successful freshwater aquatic invasive species. They're tiny, they're about 1 centimeter body length, almost transparent, and they can reach really high abundances. Originally, they're from the Ponto-Caspian region, but now they are spreading to lakes and rivers in Europe and North America. And here you can see listed 4 species that are invasive or considered invasive in the European region. So they're often found in the diet of fish, but little is known about their food web trophic effect, as mentioned earlier. So here I would like to show you the conceptual food web. And basically this is for a certain species. They're all a little bit different, as we will see, but here I'm showing you Limnomyces benedeni, which is an omnivore species, as we will see. It was found to mainly prey on Phytocera by Think et al. And to a lesser extent, they also eat copepods. And they eat rotifers, but they can also, if there's a high abundance of phytoplankton and detritus, they can also consume that. From this, we can see that the picture can be pretty complex just for this one species. So I was interested actually in what the other species might be consuming and how they would share a niche and this habitat if all of them share the same space. So here, the research question and aim of this study was that how can we quantify the trophic impact of a predator in a robust way? So we decided to use functional response curves in the laboratory for individual consumption rate of prey items. And then we used stable isotope ratios from field specimens. And then we combined the results. So at our research site in the Danube River, the 4 species are found in symmetry, suggesting niche differentiation in terms of prey and habitat preference. Here, just a quick introduction to the hauling curves. We can see the 3 different types of curves. One is completely linear with the increase of density of prey. The second reaches a plateau, and the third one changes its pattern throughout the density of the prey population. So the second idea that we need to discuss just for a quick introduction is niche space. And here we can see a diagram of a multidimensional niche space. It relies on Hutchinson's idea from 1957 that it's an n-dimensional hypervolume, and many environmental and biological axes determine a niche, but these are realized by constraints. And as Newsom and Jackson and all provided earlier, that the trophic components are an integral part of a niche. They can be quantified as the isotopic niche of an organism, but the isotopic niche is part of, but it's not the same as the ecological niche. So in this sense of our study, I would like to focus on simply the isotopic niche. The isotopic niche can be depicted here on 2 axes. On the x-axis, we can see the carbon isotope, C13, and on the y-axis, we can see the nitrogen isotope. And these 2 isotopes give us different information sources. So from the carbon, we can tell the carbon source basically, is it more open water or benthic, the source of the carbon, or from the nitrogen, we can say we can determine the trophic position of an organism. And here, these were basically the 2 methods that we combined. The data collection took place, like I said, in the Danube River, where these 4 species co-occur. And we also collected their prey items. We took them back to the lab and sorted them under microscopes. And then they were measured and weighted and freeze-dried the samples and sent to a specific stable isotope analysis laboratory. And for the functional response study, we had 200 milliliter jars filled with Daphnia pulex, and the consumption was recorded that way. So let's look at some of the results. Here we can see the stable isotope results. On the left, we can see the biplot of the carbon and nitrogen. And we can see that in color, those are the 4 species indicated by different colors, and they're found at the highest trophic position. And a little bit lower, we can see copepods, kind of in the middle. Underneath, we can see cladocerans, which makes sense because they are herbivores. And at the very bottom, we can see detritus. So if we compared the results with Poshog-Dunn's test, just to compare the niche overlaps of these 4 invasive species, we can see that only one niche overlap was significantly different, which was Hemimesis anomala, the one in red, from Catamisis varpachowskii, which is in green. So these 2 were significantly different isotopic niches, considering that these probably prey on different items. This is coming from the stable isotope results, and let's see now what we can find from comparing the 2 methods directly. So the functional response curves are found on the left, and here, just like the stable isotope results on the right, we can see that Hemimesis anomala is at the highest feeding rate, so it consumed the most Daphnia pulex, while Catamisis varpachowskii consumed the less. And the 2 other species, Limnomysis benedeni and Paramysis lacustri, are found in the middle. And this really nicely corresponds to the stable isotope results, which can be seen that, for example, the orange one, Paramysis lacustri, has a huge variation in isotopic space, and so does Limnomysis benedeni as far as the nitrogen axis. And the other 2, the red and the green, they separate nicely. So what we can say in conclusion that the 2 methods are in agreement. Basically, Hemimesis anomala proved to be the most effective at capturing zooplankton and has occupied the highest trophic position. Contrary to our expectations, the largest species, Paramysis lacustris, had an intermediate predatory efficiency and trophic position, similar to the Limnomysis benedeni. And the smallest species, Catamisis varpachowskii, is found at the lowest trophic position and had the smallest feeling rate. So what we can also see is that the 2 mostly omnivorous species had greater variation in both isotopic signatures. Along the carbon axis, the 2 pelagic species separated from the 2 benthic species. So in conclusion, we recommend using multiple methods, which might allow robust conclusions to be drawn on the trophic ecology of the species. And there's always further more work to be done. So more data is needed to map the trophic impact of omnivore species, for example, Limnomysis benedeni, which can be important in stabilizing food webs, while more predatory species like Hemimesis anomala could have top-down control. I wanted to thank my advisor, Dr. Peter Borza, for his help with this research, my university, and also funding from new National Excellence Program. At the bottom, I copied in the link to the paper, which will be published soon if you wanted to read more about the results. And with that, I wanted to thank you for your attention. If you have any questions, feel free to contact me via email at kati.patonai@gmail.com. Thank you so much.
Hello, everyone. I'm delighted to present our study on individual-based modeling of non-photochemical quenching in cyanobacteria. This study was part of my PhD at Griffith University under the supervision of Professor David Hamilton, Dr. Amir Tamad Shahidi, and Dr. Fernanda Helfer. In addition, Professor Michael Pace from the University of Virginia and Professor Kaylin Curry from Virginia Tech collaborated with us on this study. I'd like to take this opportunity to acknowledge the traditional owners of country throughout Australia and express my gratitude to Griffith University, Australian Research Council, NSF, DHI, and the Cascade Project for their support. We use process-based models to better understand the key physical and physiological processes that control cyanobacterial blooms or cyanohabs. Two types of process-based models can be used to simulate cyanohabs: Eulerian or population-level models and individual-based models or IBMs. In population-level models, bloom-forming cells are lumped into a single seed variable. On the other hand, in IBMs, each cell with a set of attributes and behaviors can be simulated separately. In addition, IBMs have the capability to incorporate antecedent environmental history and adaptive physiological traits of individuals, which provides a significant advantage over population-level models. In this work, a novel mechanistic individual-based model for the Cyanobacterium dolichospermum was developed, and non-photochemical quenching or NPQ, growth, respiration, and transport of dolichospermum were incorporated into the model. NPQ is a mechanism which protects cellular photosystems from high light intensity, and for the first time we incorporate this mechanism into a process-based model. Our study site was Peter Lake, which has an area of 2.7 hectares and a mean depth of 5.7 meters. In 2015, the lake was enriched with additions of inorganic nutrients. As a result, a bloom formed. Nutrient addition was halted on day 180 of the year, and the bloom collapsed. Our modeling system was applied to a 7-day period of this study, as indicated by the shaded area. Our modeling system consisted of a lake model and an IBM. The lake model was first run under the atmospheric and discharge forcings. Then the IBM was run under the outputs of the lake model, including water elevation, velocity, diffusivity, and temperature. The IBM consisted of 3 submodels to simulate dolichospermum growth, respiration, NPQ, and transport. The growth model of the IBM considers the effects of maximum growth rate, respiration, and light and temperature limitations. The maximum daily growth rate of dolichospermum filaments was size dependent. In this study, the model did not incorporate the effects of nutrient limitation, since the lake scale experiment caused a nutrient replete condition. Since light exposure history impacts the magnitude of NPQ in cyanobacteria, the NPQ model was developed based on the cumulative light dose each dolichospermum filament experienced. In the IBM, a high cumulative light dose led to a higher rate of NPQ and finally NPQ development in cyanobacteria, while a low cumulative light dose resulted in a negative rate of NPQ or NPQ relaxation. The modeling system was calibrated against observed data. Based on the measured and modeled water temperature profiles, Schmidt number, which represents water column stability, was calculated. As you can see, the hydraulic model was able to capture lake stability during both stratification event and the mixing period after day 180. The IBM was also calibrated against cyanobacteria cell counts observed at the depths of 75 centimeters. As you can see, the model captured the increase in cell counts during stratification event due to a higher cyanobacteria growth rate. But there was an overestimation in modeled cell counts during the mixing period. In addition, the IBM reproduced light-induced fluorescence quenching during midday. The figure on the right-hand side also shows the variation in the MPQ-free oligospermum cell counts. The maximum difference between MPQ-impacted and MPQ-free cell counts observed around noon due to high MPQ in cyanobacteria. The table shows that the incorporation of MPQ into the IBM improved predictions of cyanobacteria cell counts. This slide shows spaghetti plots of filament distribution, cumulative light dose, and MPQ at the individual level. Most filaments accumulated near the water surface during the stratification period. As a result, they experienced a higher level of cumulative light, which led to more significant MPQ during the stratification period. However, the mixing event redistributed the filaments to deeper depths. Therefore, the filaments experienced lower light and in turn lower MPQ with a shorter relaxation duration during the mixing event. Thank you for your attention.
Hello, everyone. Welcome to this talk as part of the Amplifying Voices in the ASLO 2024 conference. And my talk is on the effect of leaf litter diversity and microbial pre-processing on decomposition and fine particulate organic matter in streams. My name is Maureen Yagon, and I'm presenting this work on behalf of my team from Water Cluster Lunds and Boku University in Austria. And as you can see in that nice picture, this is the basis where we did our study, and this is a small stream, a headwater stream in Lower Austria. So as a way of introduction or introducing our work, I tell you about how terrestrial organic matter that most of the time find their way into streams are processed. So once this organic matter, which most of the time are comprised of leaf litter, are processed either by leaching and therefore releasing nutrients into streams, or are also colonized by microbes or the microbial community, which then through the process of leaf pre-processing, make the leaf more palatable for the insects that feed on these leaves. So the insects feeding on these leaves are known as shredders, and these shredders eat the leaves or preferentially feed on those leaves that have had the establishment of microbial community. Once they feed on these leaves, they produce feces, but also when they feed on these leaves, they produce small fragments of leaves. And therefore, this all component form part of the fine particulate organic matter in streams. Fine particulate organic matter in streams is very important, but also another thing that we need to consider is that once these leaves fall into streams, they either fall in sections where they are free flowing water, or like there is so free flowing water sections means the leaves that fall into these streams are exposed to oxygenated conditions. However, sometimes these leaves can fall in packs or therefore pile up in some sections of the stream, and sometimes these leaf packs can be buried in sediment. This therefore introduces another aspect of pre-processing before they are fed on by the microbial by the insects. So therefore, there is this contrast of either the oxic versus the anoxic preconditioned or pre-processed leaves before they are fed on by the shredders. So as we've described briefly, we see that there are two pathways. There's the pathway that is mediated by the microbes, but also there is the pathway that is mediated by the shredders. They work hand in hand, but this work mainly will focus on the pathway that is mediated by the shredders. So this is a conceptual diagram of what happens when leaf litter fall into streams and how they are processed until there is production of fine particulate organic matter. But what is the gap in knowledge? So we know that is climate change, but we also know that there is human activities, and these are most likely to affect biodiversity in streams. In this case, in terms of the resources falling into the streams, but also in terms of the insects that fall into the streams. However, what we don't know, or that less has been done, is the F-form dynamics, because most of the studies focus on the coarse particulate organic matter and very few actually focus on the fine particulate organic matter. So this study focuses on what happens when the shredder-mediated activities act together with the pre-processing of these leaves to influence the ecosystem functioning, in this case, leaf litter decomposition in streams. So with this, our question was, how does the leaf litter diversity and the microbial pre-processing influence the whole process of leaf litter decomposition in streams? Our hypotheses were two. And the first one was, we hypothesized that with oxic pre-processing, there is the establishment of a more diverse microbial community, and therefore there will be more higher decomposition rates and higher consumption rates, as well as the growth rates of the shredders feeding on these leaves will be higher. When we look at the F-form dynamics, we hypothesize that there will be more F-form production and also larger particles produced when the shredders feed on high quality resources. Remember, we said this leaf litter falling into the streams are of different qualities. So how does the quality influence how it is being processed? So how did we do this? We did this in the lab, where we took 3 leaf litter of different qualities, and this quality was assessed using the C-N ratios, and the good quality leaf in our case was alder, which had low C-N ratios, as you can see in the picture or the table in the bottom left. We also mixed this leaf litter so that we had treatments that had either single leaf species or in mixtures. And then this leaf litter were either pre-processed in oxic or anoxic conditions. So in the oxic conditions, we placed the leaf litter for 7 days in oxygenated water, while in the anoxic conditions for 28 days, we kept this leaf litter or leaf litter treatments in anoxic conditions. We buried them in clay. So at the end of the pre-processing or the preconditioning of this leaf litter, we fed them to Sericostoma, which is a shredder. And then we put them in microcosm, as you can see in the picture in the top right, and we let them feed for a total of 21 days. To answer our hypothesis, we measured decomposition rates. We also measured the growth rates and relative consumption rates, as well as the F-form production rates and particle sizes of the F-form at the end of the experiment. So what did we get? First, we saw that in the leaf mass loss, which is a measure of how much was broken down, alder, which was the good quality resource, had the highest leaf mass loss, and beech had the lowest leaf mass loss. And then we compared the mixtures. In the mixtures, we saw that the good quality resource was consumed first. And then upon completion, the shredders then shifted to the lower quality resources. When we look at the decomposition rates, we'll see the effect of the oxic versus anoxic preconditioning, but also the effect of high versus low quality resource. So oxic preconditioning was, there was higher decomposition rates in the oxic pre-processed leaf litter compared to the anoxic counterparts, but also the alder, which was the good quality resource, had higher decomposition rates, which is what we predicted. When we look at the mixtures did better than the low quality leaf litter. However, alder performed or had higher decomposition rates compared to even the mixtures. When we look at the Sericostoma growth rates, this was reflected in what happened in the decomposition rates. So wherever they ate, they also used it to grow the tissue. So there were higher growth rates in the leaves that had higher decomposition rates, higher growth rates in the treatments that had higher decomposition rates. When we look at the consumption rates as well, there was the strong effect of preconditioning or pre-processing, where the oxic pre-processed leaves had higher consumption rates while compared to the anoxic counterparts. Also, we can see that mixtures had very high consumption rates, almost at the same level as the ALDA, which is the good quality resource, and were very significantly different compared to the low quality resource, in this case, beach. But when we look at the FPOM production, we saw that there were no significant differences across treatments. But when we look at the marginal differences, ALDA produced more FPOM compared to beach. And you can see also that mixtures had relatively higher FPOM production compared to the beach treatment, which is the low quality resource. However, when you look at the particle size distribution of this same FPOM, we see that the good quality resource, in this case, ALDA, produced large size particles, while the low quality resource, which is in this case beach, had low size particles. What does this show? It shows that shaders, when they are given, the same shader, when given food resources of different qualities, can influence how much or how slopey they can feed on the resource. So they exhibited as slopier feeding when they were given good quality resource. How can this actually be explained? So we suggested that with good quality resources, the animals feed fast and therefore tend to produce larger particles. However, when given low quality resource, they feed, they take small bites, and therefore the resulting FPOM will be smaller compared to when they are fed on good quality resources. And when we mapped this with the FPOM weights, we saw that the smaller the particle sizes, the less the FPOM that they produce. And the larger the particle sizes, the higher the FPOM weights that they produced in the end. And if we want to look at the pictorial representation of how these particle sizes look like, you can see in that picture. And the bigger sizes are from the ALDA, which is the good quality resource. So what does our study conclude? We conclude that there were higher decomposition, consumption, and growth rates in the auxic pre-processed leaf litter. And therefore, this shows the history of leaf pre-processing really influences how much it is processed further in the chain. But also, we see the influence of mixtures. We see that mixtures actually did better when it comes to decomposition compared to the single low-quality litter. However, ALDA, which was the high-quality litter, really had higher decomposition rates even compared to the mixtures. When we look at the FPOM production and particle size distribution, we see that FPOM particles was bigger in the higher quality resources, but also there were more FPOM produced from the higher quality resources. So what does this mean? It shows that the quality of the litter really influences how the particle size distribution of the FPOM. And these have great implications on the transportation of this FPOM downstream and further processing by communities that utilize this FPOM, for example, the filter feeders, for example. So with this, I come to the end of my talk, and I want to thank you for listening. I thank my co-authors and the labs where I did my research, and also where I got my funding, and the affiliated institutions, including WASA Cluster Rooms, Boku University, and also the HR21 Doc School of Boku University. Thank you so much for listening, and I welcome questions or suggestions.
A warm welcome to everyone, to all of you, depending on wherever you are on the world. My name is Pratiksha Acharya, and today on behalf of my team, I'm going to present our work entitled Leaf Species and Preconditioning Effect on Microbial Activities and Community Composition of Fine Particles Produced During Leaf Breakdown in Streams. Going directly to the topic, leaf litters, as we know, are very important link between two different ecosystems, that is aquatic and terrestrial, which sustains different kinds of life forms and functioning in the headwater streams and plays important role in carbon cycle. When we deep down into this breakdown process, there are so many steps and several processes involved, and also the different kinds of biodiversity plays a different role during this process. As we all know that leaf fall in the autumn don't fall directly in the running water. Sometimes they are not that lucky enough and fall into the sediments where they buried for a long time and covered with the sediments. And then different kinds of microbes colonize over them. So this different oxygen level where the leaves fall in and the microbes colonizing process, this is called preconditioning. During this process, microbes really makes the leaves very nutritious and make it available to the shredders. Then shredders play a very important role in the breakdown process by their sloppy feeding and activity of gut passage. They produces two different kinds of fine particulate organic matter. And further, microbial community colonize on this fine particulate organic matter and further assist the decomposition process. When we look on the leaf litter decomposition process, we always find enough resource focusing coarse particulate organic matter and how the litter quality, shredder diversity, shredder density affects the leaf litter decomposition process. But what about these different kinds of F-form produced during this process? If these different factors also influences the same way, like it influenced to the coarse particulate organic matter? And what about the microbial contribution, which contributes highly in the coarse particulate organic matter? But what about microbial contribution on F-form is a big question. And to address these research questions, these knowledge gaps, we formulated the research question is, do different leaf species and their preconditioning affect the F-form attached microbial activities? And we hypothesized that oxic preconditioning result in higher microbial respiration on F-form than anoxic preconditioning. We also formulated our second hypothesis as do preconditioning and or gut passage influence the F-form attached microbial community composition. And we expected that preconditioning and gut passage do change the F-form attached microbial community. To test this research questions and hypotheses, we conducted microcosm experiments in the lab for which, based on the predetermined CN ratios, we selected elder as a high quality leaf litter and beech and maple as a low quality leaf litter. And then we preconditioned them in oxic and anoxic condition. After that, we feed them to Sericostoma as a shredder species in these different kinds of laboratory setup, and especially inside this magical bucket by which we get our two different kinds of fine particulate organic matter. One is shredded leaves that did not pass through the gut of the insects, and we obtain it as a sloppy feeding. And another one is fecal pellet, which went through the digestion process of these insects, and we obtain it as a leftover out of this shredder's activity. We conducted this experiment from middle of September to the first week of November on 2022 in two halves. First half covers the preconditioning process, and the second part is covers the feeding experiment where we feed these differently preconditioned leaves to Sericostoma larvae and collect a different form of F-bombs. Now, let's look what we get in our result. Going directly to the microbial activities result, we found that microbial activities is higher in oxic preconditioned leaves, irrespective of the leaf types. When we look on the fecal pellet result, we found the similar trend that we found in the leaf species, that is higher microbial respiration in the fecal pellets obtained by feeding the oxic preconditioned leaves. However, this trend of higher respiration in fecal pellets obtained by feeding oxic preconditioned leaves was vanished in the samples that we collected after 14 days and after 24 days. It might be because we did not keep up this different oxygen level during our experiment, and maybe microbes adapted to the experimental conditions. Going to the microbial community composition aspect, we see the clear differences in the microbial community composition in differently preconditioned leaf at the beginning of the feeding experiment, which is right after the preconditioning process. When we look on the leaf samples again, but at the end of feeding experiment, which was after 25 days, we saw there is not so much gap between oxic and anoxic preconditioning. However, there is still clear differences in the community composition. When we look on the fecal pellets collected at different time points, we still see the distinct differences of the oxic and anoxic preconditioning. However, also, when we look on the bulk of leaf species and fecal pellet species, we find clear differences in the composition of the microbes on our produced apome. This brings me to the conclusion of my research. And going back to my first hypothesis, yes, we find higher microbial respiration in oxic preconditioned leaves. This also persists true for the fecal pellets, which was collected after 4 days of feeding experiment. However, for the fecal pellets collected after 14 days and 24 days, it was not true. When we look on the second hypothesis, it also holds true. We found clear changes in the apome-attached microbial community composition because of the preconditioning and gut passage. This brings us to the to the take-home message, like microbes really rule the decomposition process of leaf litter at different stages, and in our case, different oxygen level and microbes on these different conditions. That is, we have defined as a redox preconditioning on original leaf litter affects the apome-attached microbial ecological processes in the streams. Thank you very much for listening. For the questions and queries, please drop the mail on the email address pop in the screen. I would also like to thank Wasserklasse Lunds for the funding to organize this amazing research, and also to all those helping hands without whom this experiment would not have been possible. Thank you very much.
Hello everyone, my name is Zora Zahir. I'm a PhD candidate from University of Regina, Canada. And today I'm going to tell you about a bit of a project of my PhD that trying to answer this question. Do environmental factors impact mercury methylators microbe in prairie wetland ponds? But before that, I want to tell you that every year more than 2,000 tons of mercury emitted worldwide, and it can be transported through air kilometers all the way to 100 kilometers away from it where it was emitted, and can be deposited on land and water. Once it's deposited, it can be transformed to one of the most toxic species of mercury, methyl mercury, CH3HG. And this is very important because it's one of the compound that is known as a neurotoxin, and it can bioaccumulate through the food chain, and human being mainly can be poisoned by methyl mercury through consumption of seafood and fish. But the interesting part about methyl mercury is that almost all of the methyl mercury in nature produced by microbes, even they have a specific two genes for it, a GCA and B. And the main function of these genes is to grab mercury and make methyl mercury. And also on the other side of this equation, there are some microbes that do the opposite. They demethylate methyl mercury, and they have also a cluster of genes, MER operon, that grab methyl mercury and demethylate it. Although we know about these microbes and this gene quite recently, but we don't know what environmental condition might enhance or inhibit this microbe to methylate or demethylate methyl mercury. To answer this question, we went to the field. Here, our site is in the middle of Saskatchewan. It's a part of 36 territories, which is a home of many Indigenous communities. But also it's very important to say that it's a part of a bigger region, Prairie Pothole region, this green area, part of Canada and US, that it's a very historically important, historically mean 10,000 years ago, and that how this Prairie Pothole created. I can tell you a lot about them, but let me show you a video that can speak a lot about the nature of these ponds. It is an expansive area of northern Great Plains. It contains millions of shallow wetland ponds. And even here in this tiny research area that we have here, we have about 100 of wetlands. These wetlands existed because of 10,000 years ago when Wisconsin glaciation started melting. It created, it started melting. That melting event was not even, and then it created this uneven depression. And very interestingly, these ponds, as you can see, they are not uniformly the same. So some of them are permanent, some of them are temporary, and also some of them hold water for 2 to 3 months. Their geochemistry is also different. But one of the characteristics of these ponds is that they usually have high dissolved organic carbon and sulfate. And mainly is that it seems that wetlands are a hotspot for mercury transformation. Kind of makes sense for us to study these wetlands. Very interestingly, in the past 2 decades or so, our lab researched these wetlands extensively, and what it was established is that there is a correlation, positive correlation, between DOC, sulfate, and methyl mercury. But what it was more interesting was that these ponds that you saw very, very, very close to one another, but their concentration of methyl mercury is different. And that's how all our researchers started to use this area as a live lab. We have so many different scenarios of geochemistry that we can test our hypothesis. So what are the main parameters that may impact microbial community and ultimately methyl mercury production? For this reason, we used this method to understand the geochemistry. We grab water sample to test for total mercury, methyl mercury, sulfate, and dissolved organic carbon, and also the routine test for water, which consists of oxidation reduction potential measurement, pH, temperature, and conductivity. And to know the microbial communities, we use a top 2 centimeter of the sediment to extract DNA and use it for 16S RNA gene sequencing, which kind of tells us that what kind of microbes we have and relatively by how much. The first thing that I wanted to do a PCA analysis, which tells me that is this ponds that we are choosing for our study are different in geochemistry. So that this PCA analysis just tell us that the ponds that we're having a very high concentration of dissolved organic carbon and sulfate kind of cluster together. The ponds with the low or kind of like medium DOC and sulfate on a cluster together. And then we had this one agricultural reservoirs locally known as dugout that it was at least is 50 years old, and I think since 1967 it was not being used, but again it's not a natural pond, and luckily happily, it was also clustered together. And this pond, this dugout, has the lowest amount of dissolved organic carbon and sulfate. What about their microbial community? Is the microbial community also kind of like clustered together and they are different in each one of these pond types or not? So for this reason, I did an ordination analysis. In ordination analysis, very similar to PCA, if the samples here, for example, these triangles, if they are clustered together in one side of the ordination, means that the microbial community composition of these samples probably are very similar to one another. And if they are not clustering, means that they are different. So here, these samples are from ponds with the highest sulfate and dissolved organic carbon, and they kind of like cluster together. Then on the other side of this ordination, these blue dots, they are ponds with kind of like moderate sulfate and dissolved organic concentration. And they also like kind of like showing a separation from the ponds with highest. And then the dugout, or that ponds with the lowest sulfate and dissolved organic carbon, like completely separated itself from the other ponds. But something that also I added in this ordination is these arrows that are showing the geochemistry ordination, how geochemistry might be related to microbial communities. So here, for example, conductivity, dissolved organic carbon, and sulfate was very close and positively correlated to the ponds with the samples from ponds with highest. And then the other side, these two ponds were correlated with temperature and pH and negatively to all other geochemistry, which was very interesting that already we are seeing a heterogeneous scenario in these ponds. What about mercury methylators? So the whole goal was to know about these microbes, that how environmental factor might affect them. So based on the literature, I grabbed these microbes that might be methylator and demethylator and categorize them based on this, and use this kind of a little bit complicated analysis to tell me how environmental factor might impact them. What I want you to grab to understand from this one is that first, there is a correlation between methylmercury, DUC, and SO4, a positive correlation, and a negative correlation with temperature, conductivity, and pH. And then these are all the correlation between each geochemistry. And then on the other side, you have methylators and demethylators that's showing that if they are weakly or highly or negatively correlated with each one of these geochemistry. So here it seems that methylators are strongly correlated to conductivity, to to sulfate, and also to dissolved organic carbon, but they are not that much correlated with the concentration of, for example, methylmercury, temperature, or oxygen reduction potential. On the other side, demethylators are very similar with the distinctions that their correlation with sulfate is weaker than methylators. With that, I want to just kind of say that we are on a direction to kind of answer this question, that do environmental factor impact methylators in peri-wetland ponds? I want to say yes, although we need way more investigation in these wetlands, and hopefully in the next few years, we will know more and more about this process. With that, I want to say thank you so much for listening to my presentation. Have a good day.
Hi everyone, my name is Miracle Denga, and today I'm going to be presenting my undergraduate research on the influence of lake morphometry on hypoluminescent oxygen demand and deep water oxygen depletion. Thermal stratification separates atmospheric oxygen from dissolved oxygen in the hypolumium. This cutoff results in a finite amount of oxygen in the deep waters for the duration of the summer stratification period. This oxygen stock is consequently depleted through processes such as aerobic respiration, decomposition, and microbial activity. Oxygen is integral to lake ecosystems as it controls habitat availability as well as several redox processes in the sediment and the water column. So with the importance of oxygen to lake ecosystems at my mind's front, my research question became, what factors affect the rate of oxygen loss from the deep waters of lakes? My three hypotheses were that morphometry, sediment phosphorus concentrations, and hypoluminetic temperature would all affect oxygen depletion rates. Predictions were that shallower lakes would lose oxygen at faster rates because more of the water column would be interacting with the sediment. For the next two hypotheses, I predicted that increases in both phosphorus concentrations and temperature would lead to faster depletion rates because they would favor greater respiration. To test these hypotheses, 19 lakes in south-central Ontario were profiled over the summer using a multi-channel profiler that recorded data on oxygen, temperature, and conductivity as they varied with depth. Sediment samples were collected at the midsummer mark from the maximum depth point, and water samples were collected in the late summer period from 1 meter above the sediment at maximum depth point. Both the water and sediment samples were analyzed for phosphorus, nitrogen, and carbon, in addition to other trace elements. To collapse the variability in nutrient results, however, their principal component 1 scores were extracted and used as a composite lake productivity variable. The depletion rates were calculated in R as the change in oxygen mass over time, both for the whole hypolimnion and for 3 meters above the sediment. With both depletion rates as the response variables and the water quality, morphometric, and physical-chemical variables as predictors, a partial least squares multivariate regression was used to describe the relationship between these response variables and the predictor variables. Our first response variable had a normal distribution and a median value of 0.25 grams of oxygen per meter squared per day. It ranged from 0.13 to 0.387 grams of oxygen per meter squared per day, a threefold difference, and these are low relative to other studies, but to be expected as they are typical for oligotrophic lakes. The second response variable had less of a trend and a median value of 0.044 grams of oxygen per meter squared per day, and this fraction had a larger range between the minimum and maximum, with a 7-fold difference between the two. So here's the first result for the PLS output of the whole hypolimnion, and our predictor variables are ordered with productivity first, followed by 9 morphometric variables, then lastly temperature on the far right. The strongest predictors, noted with the dark blue color, were the morphometric variables, particularly those associated with depth. All of these were positively correlated with dissolved oxygen depletion, and this could be because, by probability, they're more likely to be holding respiring organisms in that space. Contrary to my hypothesis and previous studies, temperature was not an important predictor of depletion rates in these lakes, possibly because the temperature ranges weren't large enough for the model to sift through. So these are our results for the whole hypolimnion. However, when we examine the benthic fraction, there is a dramatic shift in the predictor-response correlations. So now looking at the benthic fraction, we see that our strongest predictors are still the morphometric variables. However, this time they're negatively correlated with depletion rates. And this could be the influence of light penetrating into the hypolimnion and hence increasing productivity and respiration and depleting more oxygen. This study highlights the scale-dependent effects of these predictors on oxygen depletion rates. The morphometric hypothesis was well supported at both scales, and has been found to be a consistent predictor of oxygen depletion and anoxia for low nutrient lakes, as seen in studies by Bukovacs et al. And Deeds et al. Lake productivity became a moderate predictor when examining the benthic fraction, and this was likely because activity in the sediment was better captured by the model when examined at a narrower constraint. Overall, however, the sediment phosphorus lake productivity hypothesis was not well supported in contrast to previous studies. These lakes are low nutrient, so have minimal variation in their nutrient concentrations. Therefore, it's likely that the model did not have enough variation to sift through. Because my hypothesis was nutrient-related, it would have been prudent to have a larger range of nutrient concentrations in the lake samples. In the future, I would choose lakes spanning a range of trophic statuses, from oligotrophic right up to eutrophic. Although we've seen that morphometry has a large influence on dissolved oxygen depletion rates, this study was only done across one summer and offers a snapshot in time. It would be interesting to see if there's any interannual variability to the predictors, and if morphometry is a consistent predictor of depletion rates, or if there could be something else in different years. Lastly, light was potentially unaccounted for with these rates, so in the future, I would take a light profile to offer a more fine-scale record of how light varies with depth. Looking to the future, it'd be worthwhile to see whether these lakes have any threshold nutrient concentrations where their contributions to oxygen depletion become important. This would be possible with ranging trophic status alterations. We see that morphometry affects the depletion rates. Could we take it one step further and predict what depletion rates would look like when given a set of morphometric descriptors of a lake? This work lays a good foundation for that effort. We would then be able to predict the effect size that a changing climate will have on these rates. Lastly, linking to climate change, it's no secret that winter is changing. What does this mean for winter depletion rates? And could they also be morphometry driven, or are they driven by something else? To conclude, this study lays the necessary groundwork for learning more about the factors that drive oxygen loss in temperate oligotrophic lakes, which has proven to be primarily morphometry driven. This work is important not only for understanding our present ecosystem health, but understanding what our lakes' ecosystems will look like in the future. Thank you.